Alright, hello 3A5. So we are here to continue our next lesson, which will take us through nicely to the end of this chapter. So let's just do a little brief refresher of what we covered the previous time. We spoke about why the small intestine is so perfectly adapted for its function in absorption. Now remember these, okay? These are what we consider to be villi, okay? And then all these crisscrosses around the middle portion. These are your blood capillaries, in which your sugar molecules as well as your amino acids will diffuse across the wall of the villus, which is very thin, so that diffusion can be fast, and then will be transported by the blood capillaries to other parts of the body to be used. Okay, so why must food be digested? So that it's small enough to pass through the partially permeable membrane. Alright, so let's continue to the next part. Alright, so today we are going to be talking about the function of the liver. So we know that the liver is actually connected to the small intestine, not directly, but indirectly by a blood vessel known as the hepatic portal vein. So you just imagine, alright, that this is your small intestine. Okay, so whatever glucose that has been absorbed into your small intestine will then be transported via your hepatic portal vein. So why do we call it a portal? Because it's in between two places. So it brings the blood directly from the small intestine towards the liver, in which the liver will do a few things, right? It will store excess glucose as glycogen, okay, and it will get rid of excess amino acid in the process known as deamination. Okay, so it will convert excess amino acid and glucose into glycogen and urea respectively. Then what happens to the rest of the other glucose that is not converted, or the rest of the other amino acids that are not deaminated? They will then be sent to the rest of the body via your hepatic vein. Okay, so this was what we covered in the previous lesson. So let's now move along. Alright, so what are the different functions of the liver? Now, the liver is a very important organ and it carries out many activities, and that is why it requires a lot of energy. Okay, so your liver actually is an organ that requires a lot of energy, and that's why all the liver cells, alright, have a lot of mitochondria. Okay, now we know what does a mitochondria do, right? The mitochondria basically releases energy. So why must a liver cell have a lot of mitochondria? Because the liver cells carries 1, 2, 3, 4, 5, 6. It carries out 6 activities, which is a lot of activities. So it requires a lot of energy, and that's why every liver cell has a lot of mitochondria. So we must be able to link the function to the structure. So if I were to ask you, how come the sperm cell needs to have a lot of mitochondria? Because the sperm cell has a tail, and the tail is about movement, and movement requires a lot of energy. Alright, so these are just some functions of the liver. It is involved in deamination. Remember, in bio, de means to remove. Amination means you are removing the amino group of the amino acid. So you're actually taking amino acid and you are converting it into urea, which is a less toxic substance. Okay. Next thing, the liver is also involved in detoxification. Again, you see the word de appearing. So in bio, de means to remove. So what you are doing is you are removing the toxins. So you are changing something from toxic into less harmful, or basically into harmless substances. The liver is also involved in the storage of iron. It's involved in making proteins as well. Alright, definitely you know what is bile, right? Bile is involved in the emulsification of fats so that you can increase surface area to volume ratio for faster digestion by lipase. And last but not least, is involved in blood glucose concentration. So previously, you would know that your liver actually converts excess. Okay, so it converts excess glucose into glycogen. And since it converts excess glucose to glycogen, what it actually does is it actually helps to control, okay, the concentration. What's the meaning of control? It means it helps to keep something constant or normal. All right, so I remember I shared with you what the liver actually does. Okay, so for example, if the liver absorbs, let's say 10 units of glucose, the liver actually has 10 units of glucose coming from the small intestine, and the rest of the body only needs 2 units of glucose, let's say. That means I only want 2 units of glucose to be inside the hepatic vein, transporting it to the rest of the body. So what the liver must do is the liver must basically get rid of this excess, but it doesn't get rid of it, it stores it for later use. So what you can see, originally I started with 10, now I only have 2 because 8 has been stored in glycogen. This is what we mean by regulation. That means you are keeping the concentration level, okay? So let's move on and continue. So I just want to show you a video, alright, that will help to do a little summary of the whole function of the liver. But let me just get rid of my annotations first. Right, so let's watch the video.
There's a factory inside you that weighs about 1.4 kilograms and runs for 24 hours a day. This is your liver, the heaviest organ in your body and one of the most crucial. This industrious structure simultaneously acts as a storehouse, a manufacturing hub, and a processing plant. And each of these functions involves so many important subtasks that without the liver, our bodies would simply stop working. One of the liver's main functions is to filter the body's blood, which it receives in regular shipments from 2 sources. The hepatic artery delivers blood from the heart, while the hepatic portal vein brings it from the intestine. This double delivery fills the liver with nutrients that it then sorts, processes, and stores with the help of thousands of tiny internal processing plants known as lobules. Both blood flows also deliver the oxygen that the liver needs to function. The blood that is received from the intestine contains carbohydrates, fats, and vitamins and other nutrients dissolved in it from the food you've consumed. These must be processed in different ways. In the case of carbohydrates, the liver breaks them down and converts them into sugars for the body to use as energy when the filtered blood is sent back out. Sometimes the body has leftovers of nutrients that it doesn't immediately require. When that happens, the liver holds some back and stacks them in its storage facility. This facility works like a pantry for future cases when the body might be in need of nutrients. But the blood flowing into the liver isn't always full of good things. It also contains toxins and byproducts that the body can't use, and the liver monitors these strictly. When it spots a useless or toxic substance, it either converts it into a product that can't hurt the body or isolates it and whisks it away, channeling it through the kidneys and intestine to be excreted. Of course, we wouldn't consider the liver a factory if it didn't also manufacture things. This organ makes everything from various blood plasma proteins that transport fatty acids and help form blood clots, to the cholesterol that helps the body create hormones. It also makes vitamin D and substances that help digestion. But one of its most vital products is bile. Like an eco-friendly treatment plant, the liver uses cells called hepatocytes to convert toxic waste products into this bitter greenish liquid. As it's produced, bile is funneled into a small container below the liver, called the gallbladder, before being trickled into the intestine to help break down fats, destroy microbes, and neutralize extra stomach acid. Bile also helps carry other toxins and byproducts from the liver out of the body. So as you can see, the liver is an extremely efficient industrial site, performing multiple tasks that support each other. But such a complex system needs to be kept running smoothly by keeping it healthy and not overloading it with more toxins than it can handle.
Alright, so I think that is a very nice video to summarize what the liver actually does. So let's move on to our next part. Okay, so the very first function of the liver, which we will talk about, and this is a very important O-level concept, is that the liver is involved in regulating blood glucose concentration. So earlier we actually said, right, that regulating, all right, regulation means to control something. All right, so it's basically helping to control blood glucose concentration, and it does so by interacting with two different types of chemical substances in our body known as hormones. The first hormone, all right, is known as insulin, and the second hormone is known as glucagon. Now, people always confuse glucose and glucagon, so this one you really have to write like a few times for you to remember. All right, so the liver is stimulated. The meaning of stimulated means it is activated. So without insulin and without glucose, the liver cannot do anything. But the moment the liver receives insulin and glucagon, all right, then it will be able to carry out its action. So that means, right, insulin and glucagon, they are not the ones who are carrying out the action. They are basically, so if this is your liver, all right, let's say insulin is represented by capital I and glucagon is represented by capital G. The moment the liver receives insulin, there will be a response within the liver. If the liver receives glucagon, there will be a different type of response for glucagon, right? So the liver is basically reacting or responding or being activated by the presence of these two hormones. And these two hormones help to control the glucose concentration in our blood. So how is the glucose concentration in our blood controlled by insulin, glucagon, and the liver? Number one, when there's too much glucose in the blood, you may want to write this down. If you feel that you need to pause the video, please pause the video. Too much glucose in the blood could be, for example, after a meal. Example, after eating a meal that is very high in carbo, because you know carbohydrates contain a lot of starch, and starch will be eventually digested to maltose and finally to glucose, right? So when there's too much glucose in the blood, what happens is that this sends a signal to the pancreas. Okay? The islets of Langerhans is found in the pancreas. So you remember the pancreas also releases the three different types of enzymes, right? LAT, lipase, amylase, and trypsin. All right, that is the digestive role of the enzyme of the pancreas. The pancreas also has another function, which it releases insulin, which is the hormone. Now, this hormone will travel from the pancreas in the blood to the liver. Now, when the liver cells receive insulin, what the liver will do is, the moment insulin is there, the liver will convert all the extra glucose, okay, all the excess glucose will then be converted into glycogen. Now, do you remember what is glycogen? Okay, glycogen is the animal form of starch. It's like starch, but it's not starch because we are not plants. So it stores glucose in the form of glycogen. Then it keeps it. Then when it needs it again, it will break glycogen to release glucose. Take for example, if you bought too much food for lunch one day and you cannot finish all the food that you have, what do? Instead of throwing away the food, you put it inside your fridge, right? To keep it so that later at night you can take it out and reheat it and eat it again. So basically the liver is acting like a fridge, okay? It will store all the excess glucose in the form of glycogen. Remember, okay? And then what happens? What happens is that the blood glucose level will then decrease back to normal. Can you please write that down? It decreases back to normal. Now, our human body actually has about 80 to 90 units of glucose that is considered to be normal. Okay, that is when you eat too much glucose. Now, what is the next situation that you might be in? For example, you may not have eaten for a few hours. Like, for example, some of your Muslim friends who had just finished their month of fasting. If not, when you have finished a period of exercise, right? That means that there is too little glucose in the body now. Now, when you have too little glucose, this is a very dangerous situation because you can have headaches, you can feel giddy, and sometimes you might even faint. So your body does not want that. So remember, your liver helps to control, right? So now if you have too little, what your liver wants to do is that it wants to increase it back to normal. And remember, the liver cannot do it on its own. It requires a hormone. Now, it does not require insulin because insulin decreases the blood glucose. Now, if insulin is released in this situation, habis, there will be even lesser glucose in your body. Therefore, it won't be insulin that will be released. However, your pancreas, whenever you see isolates of Langerhans, it means the pancreas. So your pancreas will release a new hormone known as glucagon. Okay, glucagon will then cause the liver to convert glycogen into glucose. Now, why do you want to take the stored glycogen and break it down to release glucose? That's because you want the glucose level to increase back to normal. So you see, when you take the stored food and you break it down, you are actually releasing more of glucose into the blood. Therefore, from very little, it will go back to normal again. Okay, so this slide is actually very important. So can you please make sure you put 5 stars next to this part? You might want to re-watch the video again to help yourself understand better. But in summary, just know that the liver is very important in controlling the glucose concentration because it helps to either decrease it if it's too much, or it helps to increase it if it's too little. But it cannot do it on its own. It requires activators, and the activators are known as insulin and glucagon, and these are produced by the pancreas. These are known as hormones. All right. So I just want to show you this little animation. Maybe it will help you to be able to visualize better. So this is the level of circulating blood that is constantly monitored and kept regular by the pancreas. So all these little shapes here that you see, these are your glucose molecules. Now what you see, these pink color long tubes, these are your blood capillaries that help to transport blood to and from the pancreas to the liver. Now this portion over here is known as your blood capillaries that come from the small intestines. Okay, so what happens after you eat? You know that after you eat, you will basically have an extra levels of glucose. You will have more glucose, right? Because if you eat more, then of course your small intestine will absorb more glucose, so naturally you have higher glucose. So see what happens to the number of hexagon shapes. It increases, right? Now if it keeps on increasing, then you will have too much glucose in the body. And what happens? Your pancreas will detect that, it's too much. It will release this blue molecule known as insulin that will travel in the blood to the liver. Now remember, without insulin, the liver cannot do anything. Now that there is insulin in the blood traveling to the liver is stimulated, is triggered, is activated. Okay, it's like there's a switch that is turned on, telling the liver to do something. And what will the liver do? I want you to notice what is happening on top here. You will realize that there's lesser and lesser glucose molecules. You see the glucose molecules forming a long chain? Now this is known as your glycogen molecule. So you will notice that the number of glucose molecule is decreasing, but over here you can see that the glucose molecules are joined together to form glycogen to be stored in the liver so that the glucose concentration will go back to normal again. Okay, so now what happens after fasting or after exercising when you have too little glucose concentration in your blood? So after exercising, the blood glucose is very little. You can see, right? The number of glucose molecules too little. The pancreas will detect that is too little. Because too little, I said you get headaches, you feel giddy, you want to vomit. So it will detect it and then it will release another hormone known as glucagon, which is a green triangle. So you see, now you watch what happens when the glucagon goes to the liver. This is your glycogen. You notice what happens to the length of the glycogen molecule. Okay, the liver under the influence of glucagon will start to break down and release more and more glucose molecules. So this one was from before you store it, right? So now you can see it's becoming shorter and shorter because the chemical bonds are broken. Therefore, your glycogen molecule is being broken down and released into glucose. So this is what happens, and then the glucose concentration increases back to normal again. So I hope this animation was a little bit more helpful for you to be able to understand how the liver, together with glucagon and insulin, helps to regulate the glucose concentration. Now, regulating glucose concentration is also known as assimilation because you are using the nutrient glucose after absorption. Okay? Alright, so let's move on. Okay, so the next function of the liver is in bile production. I don't think I need to go through this with you. Alright, the liver cells are the ones that produce bile. Remember, not the gallbladder. And what it does is that the liver is connected to the gallbladder by small ducts. So once the bile has been produced by the liver cells, what actually happens? Okay, once the bile has been produced, what actually happens is that the liver actually releases the bile and it travels to the gallbladder. Okay, and then the gallbladder will then release this bile directly into the small intestine via the bile duct. Okay, and you know what bile does, right? So bile emulsifies fats to increase surface area to volume ratio so that you can increase digestion by lipase. All right. So let's move along. Okay, the next function is that it actually stores iron. Now, your red blood cell actually contains iron because hemoglobin actually contains the element iron, which will help to combine with oxygen. So what happens is that the red blood cell will basically make its way to the spleen, which is another organ that is quite closely associated. And then what happens is that it will break the red blood cell down and it will release hemoglobin. Now, this hemoglobin will then travel to the liver. It will then break down hemoglobin into two things. Number one, it will basically store the iron that is inside hemoglobin. So this one contains iron. All right, so it will recycle the iron in the liver. And the next thing is from broken down hemoglobin, it forms bile. So actually that's where your bile comes from. Your bile actually comes from hemoglobin, which is from those old red blood cells that cannot be repaired, remember, because they don't have a nucleus for reproduction. So whatever that is broken down from hemoglobin will basically form bile. Okay. The next function is to make proteins, and some of these proteins that your liver makes are plasma proteins. Now, why do we say plasma proteins? Because these are found in the plasma. They are involved in blood clotting, okay, which is a very important process, which we will learn in the next few chapters. It basically helps to prevent excessive blood loss. Okay. At any point of time, if you want to take down notes, you can always pause the video. Now, the next function of the liver, which is also very important besides the regulation of glucose, is the deamination of amino acids. So we see this word again. De means remove. All right. And what do you mean by deamination? That means it's removing the amino group. Okay. So please remember, they don't deaminate all the amino acids, and it's quite useless to absorb any amino acids, right? They only deaminate the excess. That means the ones that your body may not use because it's extra. So in deamination, what happens is that the amino groups will be removed and these will be converted to urea. Now, urea sounds like, you're right, urine. Okay, so urea is basically excreted inside your urine. That's why your urine has a very pungent smell. So this is just a little chemistry behind the deamination of amino acids. All right, but you don't really need to know this. If you want to, you can pause the video and have a look at it, or you can ask me more questions later on. Right, the next third important function is detoxification. Sorry, the fourth important function is detoxification, right? That is when you ensure that harmful substances such as alcohol, okay, are broken down to give you harmless substances. Now, alcohol is broken down by a particular enzyme known as alcohol dehydrogenase. This is found inside your liver cells. So your liver cells are so important because not only do they produce bile, not only do they help to regulate glucose and amino acids, not only do they produce proteins, but they also have an enzyme that will break down alcohol to give you a harmless substance. Okay? So now, we have come to the end of the function of the liver. Now we talk about what happens when the liver is not functioning. So what are some of the situations that can cause liver failure? All right, when you take too much excess alcohol, or certain diseases that may lead to liver failure. So what happens if there is excessive alcohol consumption? Besides having gastric ulcers, it can cause liver cirrhosis. Now, liver cirrhosis is damage to the liver. It can slow down brain functions and increase reaction time. That is because alcohol is a very common depressant. A depressant means that it basically causes your body to react longer. So instead of like taking 5 seconds to react to something, you may take like 15 to 20 seconds. That's why they say don't drink and drive, because your reaction time to press the brakes or to react to something will be longer, and then you might have an accident. It also reduces self-control. That means people who are usually intoxicated or very drunk in alcohol, they tend to not know what is happening around them, and they may say things or react in ways that they can't control. Okay? Yes, and one of the social problems is that frequent intake leads to addiction. So this is a social issue. Okay? But the more important thing is, since we're studying about the function of the liver, we want to know why or how can we prevent this. Okay, basically in liver cirrhosis, your liver cells are destroyed and they are replaced with fibrous tissues. Now, fibrous tissues are basically hardened. They are hardened substances. So what it does is that it damages the cells and it prevents the cells from carrying out its normal function. Excessive alcohol can also lead to hemorrhage. Hemorrhage is bleeding in the liver, and this will eventually lead to liver failure and even death, because we know that the liver is a very important function, right? But how come alcohol can actually cause gastric ulcers? That's because it stimulates increased acid secretion. And you know what is this acid, right? It's your hydrochloric acid. That would mean that there is a lot of acid inside the stomach, and it starts to corrode, or it starts to digest away or dissolve away the mucus lining of your stomach, eventually causing the acid to burn through the walls of your stomach, which is a very painful situation that can lead to ulcers. Okay, so over here we can see a healthy liver. Now this is an unhealthy liver in the disease known as cirrhosis. It looks a little bit disgusting, right? You can see that there are a lot of bumps and it looks very rough. So all these bumps that you see over here these are your fibrous tissue, right? That's why they look like they are filled with something. Okay, so I'm just going to ask you to watch